Hey everybody, I'm Josh from USU Extension, and I'm excited to welcome you to this portion of the Agri-Science camp. Today we're going to be learning about DNA extraction and how to do it. We will also talk about some of the principles of DNA extraction and how that is applied to agriculture. All right, for today's DNA extraction method, we're going to be utilizing split peas so that we can extract DNA from them. So what you're going to need, a half cup of split peas, a cup of cold water, an eighth of a teaspoon of salt, some meat tenderizer. Then you're going to need 2 tablespoons of a liquid dish detergent, some isopropanol alcohol, a pipette, a test tube, and a Q-tip. You'll need a strainer and then also a blender. Those are the items that we're going to be utilizing today, and we're going to go through the steps on how you'll extract the DNA from the peas. All right, our first step is going to be to add the half a cup of split peas into our blender. We're also going to add a cup of cold water and then our eighth of a teaspoon of salt. Okay, and then we're going to hurry and put it in our blender. We've got these 3 ingredients in the blender. We're going to want to carefully blend them on a high power for just about 15 seconds. You don't want to overdo it or underdo it, so make sure to keep within that 15-second range. All right, and this is after the blending has been done. You can see that green, light green color. That's the liquid type of the color that we're going to want to see there. And you will have some froth in there from the blending. But here's just a little example of what it should look like after that 15 seconds. All right, now that we've got our stuff blended together, we're going to simply take it, put our strainer on top of just a cup. It doesn't matter what your cup looks like, just something to catch the liquid into. But we're going to just pour all of the contents that we have in our blender into this strainer. Okay, and then after we've strained that through, we're going to take a teaspoon and our liquid detergent. I've chosen a clear detergent, but I've also done it with other colored detergents. Whatever you have at home will work just fine. But you're going to take that and you're going to add about 2 tablespoons of this liquid detergent to your pea solution. Okay. Then what I'm going to do is I'm just going to use my spoon here, and I'm going to just lightly stir it, not very heavily at all, just lightly a couple times around, and then I'm going to leave it for about 5 to 10 minutes to settle. Our next step is going to be to pour our solution into a test tube. We're going to only want to fill the test tube about 1/3 of the way full with our solution. All right, our next step is going to be to add a pinch of a meat tenderizer enzyme into our test tube. Our next step is going to be to add our isopropanol alcohol into our test tube. And we just want to add enough that we can get a layer that will develop on top of our solution that's already in the test tube. Alcohol is less dense than water, and so it will form a separation layer on the top. Now when we pour this in using our pipette, we want to have a tilt to our test tube, and we want to run it down the side very carefully to create that layer. Okay, and I'm actually going to add just a little bit more to it, but if you look very carefully and closely, you will see that the DNA is actually visible there coming up in its strand form in that test tube, in that aqueous layer. All right, our last step in order to see the DNA just a little bit closer is we're going to take a Q-tip and we're going to come down here into this layer and we're going to just give it a little bit of a stir and kind of grab that DNA. And if you look really carefully, you can see that we have those strands of visible DNA. It kind of looks like a white snot almost, for lack of a better term, but that is what exactly you're looking for. That is split pea DNA that's hanging out there inside of that alcohol solution. All right, now we want to explain to you the science behind what we have done today in creating this extraction of DNA. So first, our P cells are something that contains DNA. And so we use them to be able to have something to extract from. We use our blender to be able to separate the P cells from each other so that we can have a really thin layer of that soupy substance with those cells that are in it. The salt comes into play because it helps to bind the DNA. Even though we use it in our one of our first steps, it in the final process is what helps us to be able to bind that DNA. Our detergent actually helps to capture the proteins and lipids of the cell membrane. And then when we are able to add our meat tenderizer, the DNA in the nucleus of the cell is molded, folded, and protected by certain proteins. And so this meat tenderizer actually goes in and cuts the proteins away from the DNA. Lastly, our alcohol, it's less dense than water, so it floats up to the top, forming that separation of layers. Alcohol also in the laboratory and DNA extraction is used for purification of the DNA sample. All of the grease and the protein that we broke up in that test tube in the first couple of steps when the alcohol is added actually also drops down to the bottom, allowing that DNA to float up to the top. So essentially, using these different chemicals and formulations, we are able to take a simple split pea and extract a great sample of DNA from it. And this type of sample can be used to be amplified, to be able to do studying with and do all sorts of different things. We can use DNA for forensics, and we can use it for the advancements of science. For me personally, I've been able to use DNA to help to create parentage analysis samples in cattle. So being able to take their hair follicles, extracting their DNA, and running it through processes that can tell me who the dad is, what bull was the dad to the calf, and also who the mom is if we're confused at the time of which calf belongs to which cow. We've also been able to use DNA extraction in pig genetic studies, where we've been able to also use that hair follicle to extract DNA and then be able to study certain genetic abnormalities in hogs that lead to poor meat quality or other issues. Again, DNA and extraction are something that we can utilize in all different aspects of our lives and utilize the science to help improve the things